everyone, and welcome back to my channel. So you may notice that something is a little bit different. We are at McMaster University and we are talking to Shawn, who is a researcher. Would you like to introduce yourself? Say hi.
Hey, yeah.
So, instead of introducing his research in the traditional way...
Really, you just say, hi, my name is whatever, I do XYZ.
Yeah, instead of asking you just the typical, tell us about your research questions, we have a few prompts that are loosely related to your research in this little bin. And you're going to have to pull it out and answer it. And
this is kind of scary because I don't know what she has in there. She could have anything in there that I probably know very little about. Hopefully I don't pull something that I don't know much about.
Yeah, let's test his knowledge.
Okay.
Let's get started.
So the first word, okay, why do science outreach? So it's very interesting that you asked this.
Yes.
Why do science outreach? So science outreach is important to me. I guess I kind of have to talk about what I do. So very loosely, I do breast cancer research in a molecular biology lab at McMaster, in Dr. Juliet Daniel's research lab at McMaster University in the Department of Biology. So generally speaking, if you're in a research lab in biology, you're like confined to your lab, you're just doing lots of experiments, testing cell lines, doing mice experiments and what's not. However, my research tends or allows me to go outside of the lab and to interact with the community. So why do science outreach? To answer that is because I don't like to feel such a strong disconnect from the communities that you're going to impact with the science that you're doing. So in my case, specifically, we do outreach in communities for Black women across Canada, well, across the GTA, I should say, where we know that there may be gaps of knowledge in this particular population. So that is why I do science outreach along with the members in my lab in these populations.
Awesome. Well, what kind of science outreach have you done?
Yeah, so the science outreach, it's more like community engagement kinds of outreach, in the sense that our lab, we partner with this partner in Toronto called the Olive Branch of Hope. And that's specifically an organization that was launched to offer assistance and support to Black women diagnosed with breast cancer. So it was founded by this lady, her name is Layla Springer, and she was diagnosed with breast cancer many years ago, and she didn't have a community to really talk about and engage with. So she started it along with another friend of hers, and basically whenever someone else is diagnosed with breast cancer, they will meet with them and they will talk with them, support them, just give them help and just let them know that there's a community. So we partnered with them, Dr. Daniel partnered with this group, and now we do outreach in the GTA and Hamilton, where we just do education specifically to young and premenopausal women of African ancestry.
Very cool.
So in that, we talk about the research that we've done, and we also talk also very broadly about diet, exercise, just living healthy lives, and not just like heavily like cancer.
Yeah.
Because it's like that's kind of a scary word for a lot of people.
Yeah. So you start at all ends of the spectrum of health.
Yeah.
Cool.
Okay, next.
Okay.
Wow, if you weren't a scientist, what would you be? I thought about this today.
Amazing. What a coincidence.
Yeah, like I thought about it actually because someone posted this on their Instagram using the question thing, and I responded instantly. Like I knew exactly what I was going to be if I weren't a scientist. So my answer today to that Instagram story would be a musician. So fun fact, I play the violin. I've been playing for 15 years.
What?
So I love music. I definitely would be a musician. I was actually going to go to Italy on a music scholarship when I was 17. Oh, I'm from Barbados. I should have added that.
Cool.
So I was gonna do that. It didn't happen. Many circumstances, it just didn't work out that way. But if I if I wasn't a scientist, I definitely would be a musician.
That's so cool. Do you still play, like, sometimes?
I do. I picked up the violin on Sunday.
Wow, that's amazing.
Yeah, no, I love music. I love the violin specifically, but like I love music. I always have music playing. There's always music subconsciously playing in my head.
You're just walking around, there's music.
There is music, like there's a soundtrack right now.
What is today's soundtrack?
It's not like a specific soundtrack, it's just like music.
Yeah. That's so cool. I did not know that. Cool.
I like to sing. I play a little piano, like I started out on piano. Yeah, I definitely would be a musician.
Jen, look out for your new album.
Yes, the Hercules album.
Do you have less research to do?
Will that ever be a thing? Okay, next question. Triple negative breast cancer. So that is what that's what we investigate in our lab, triple negative breast cancer. So what is triple negative breast cancer, I'm guessing is your question, and like whatever I want to talk about it.
Yeah.
Okay, triple negative breast cancer is defined by the lack of the expression of 3 biomarkers. They can be thought of as doors that are either like open or closed. So it's the estrogen receptor, progesterone receptor, and human epidermal growth factor receptor 2. And if the door is open, it's considered positive. If the door is closed, it's negative. So there's some tumors like the estrogen receptor door, if that's open, there are targeted therapies that can enter through that door and effectively shrink the size of the tumor. And also for the human epidermal growth factor receptor 2 or HER2 door, if that door is open or positive, present, there are targeted therapies that can target the HER2 positive tumors, and those specific therapies have led to increased survival in patients that are positive for either one of those tumors that are positive for those receptors. However, triple negative breast cancer are negative, so they're like the doors are not present; they're closed. For the ER, estrogen receptor, progesterone receptor, and HER2, so ER, PR, and HER2. So there are no targeted therapies for those specific tumors, so therefore the survival is typically lower in those tumors. And not only that, these tumors have a higher propensity to spread to other vital organs, such as like the brain, the lungs, the liver. So that's also another issue. Another compounding issue is the fact that even though there's a higher incidence in the US and the UK of breast cancer in white women compared to black women, there is a higher mortality rate in black women for breast cancer. And we're not sure why that is. Some people say it's due to social economic status, other lifestyle factors, knowledge about mammography, like all of these other factors. But even after adjusting for those things mathematically, we still see that there's a higher mortality rate from breast cancer in black women. Yeah. And specifically, triple negative breast cancer, there's a higher prevalence of triple negative breast cancer in black women and Hispanic women. So we're thinking that this high mortality rate in breast cancer has something to do with the fact that there's a higher prevalence of triple negative breast cancer in that population. So that is everything I can talk about right now about triple negative breast cancer. But any other questions you might have about triple negative, you can ask away.
So mortality rates might be higher because triple negative breast cancer is more common, which is a more likely to lead to mortality kind of cancer. Yes,
it's just a more aggressive cancer overall. Yeah.
It's interesting. It's so interesting. It's interesting that you're learning something that has never been, like, is unknown so far.
Yeah.
Which is a lot of what novel research is.
Yeah, so there are lots of researchers across the globe trying to understand triple negative breast cancer, seeing if there's potentially another door that we can open up to target triple negative tumors. But yeah, there's a lot to be done.
Cool.
With that.
Very cool. Some of the other cards might relate to that as well.
Ooh.
Let's see.
The protein KAISO. Okay, so KAISO was identified by my supervisor, Dr. Juliet Daniel, while she was doing her postdoc a couple years ago, actually almost 20 years ago now. Wow. So KAISO was identified, as I said, by Dr. Daniel, as a binding partner to P120 catenin. I'm getting a little too scientific here, I think. But
what does that mean?
But KAISO, specifically, its expression has been linked to the aggressive nature in various cancers. So there's prostate cancer, colorectal cancer, pancreatic ductal adenocarcinoma. So there are various cancers in which KAISO has been implicated in some way. Specifically in breast cancer, we see that, and other cancers actually, we see that KAISO's expression is linked to the ability of the tumor cells to metastasize to various organs. So in cells where there's high KAISO, there's also higher expression of other biomarkers that are for metastasis, for the cancer to spread. And we also see specifically in our populations of women of African ancestry that there's higher KAISO expression in women of African ancestry compared to Caucasian American women with breast cancer and also prostate cancer. So it's really interesting. Not only is KAISO playing a role in the aggressiveness of various cancers, it's also playing a role in the racial disparities that we see for at least breast, prostate, and pancreatic ductal adenocarcinoma. So it's interesting. Lots of work to be done with KAISO as well, because who knows, KAISO could be that other door for the triple negative.
Is KAISO something that you test for? Do doctors test for that?
No, but we're hoping that maybe one day they will, because it may be a diagnostic tool, but there's so much work that has to be done to like get anything to that step. Yeah. So that's why like a lot of the groundwork is being laid down now by everyone in the lab, all of the undergrads, the other graduate students working day and night.
Cool.
About KAISO.
Very cool.
Yeah.
Very cool.
Next.
Yeah, next.
Okay. Why mice?
You do research on mice.
Yes, the lab does research on mice.
Yes.
So why mice? Because they are a relatively easy model organism.
Yeah.
And we're a lot genetically identical to mice.
Do other cancer labs and other universities perhaps study different model organisms?
Yeah, there's so many model organisms. Yeah. But mice specifically is like kind of like the first step that you do. And
then if you want to have a therapy to reach humans, you have to go through many steps, and mice is probably the first one.
Lab procedure.
Tell us about what you do in the lab. Or what is your lab procedure? What are some of the equipment?
There's so many things that I can talk about for lab procedure. There's so many procedures.
Yeah.
Okay, so in our lab, we do a lot of immunoblotting, a lot of immunohistochemistry. We do a lot of work with, so my project is using immunohistochemistry and also sequencing. My project also has some epidemiology as well, but generally the lab is more of a molecular biology kind of lab. So procedure, I'll give you an example of one procedure. You can ask me about any other procedure also. But immunohistochemistry for my project. So earlier I talked about the fact that there was higher expression of KAISO in the women of African ancestry tissues compared to the Caucasian American tissues, and we used a procedure called immunohistochemistry. So essentially, we collected tissues, we went through rigorous ethics approval from the various institutions, and we collected breast tumor specimens from women in Nigeria and Barbados, and we constructed what you called a tissue microarray. So we had a pathologist that selected certain parts of the tumor specimen and selected it from various patient samples and place it onto one block. And then we take that block and we get one microscope slide with all of these samples. And then immunohistochemistry is a procedure that we use to identify expression and localization of various proteins. So essentially, there's an antigen that's present on the tumor, on the sample, sorry, and you use a primary antibody that will bind to that antigen, and then a secondary antibody that will bind to the primary antibody that's bound to the antigen, that will emit a signal that you can then visualize. So then if the primary antibody is bound to the antigen, that means that the antigen is first of all there. So if we see that it's more highly expressed, we know that more primary antibody was bound to the actual antigen. So that's one technique I can talk about.
And it's all a way that if it's not present, it'll wash away and you won't see the signal.
Exactly. So if it's not bound, there'll be no signal. Or if there's less of it bound, there'll be less signal. Yeah. Kind of thing.
I feel like a lot of biotechnology research is very complicated with like this binds to that, binds to this, but it's all kind of trying to detect something that you can't see with something that you can see.
Exactly.
So
that's exactly what immunohistochemistry is. And immunoblotting. The sequencing is it's not like that in any way, actually. But we take the samples, we use the same samples, and we extract DNA from the samples. It's possible, it's very challenging, but it's possible to extract the DNA from these samples, and then you'll be able to sequence them and see if there are any changes between the tumor tissue and the normal tissue, and see if, like, see what mutations are actually driving the tumor. Yeah.
Very interesting. It seems so complex, but in an everyday situation, you're just kind of like running a standard procedure and then waiting for results.
Yeah.
How long would you say you collect results for to find?
I'm starting the 4th year.
Cool. Yeah.
Of this journey. So I've been collecting results for now almost 4 years. And I will be collecting results for a little while longer. And then you would use the information you have so far to come to a conclusion so far. Yes. But then you could continue to do research to hopefully find more. Yeah. So the idea is that you'll be publishing throughout the entire time. So I'm actually drafting 2 papers right now based on what we found so far. Yeah.
Very exciting.
Yeah. Favorite thing about science? The fact that I can do this. Favorite thing about science? I am just a very curious person. Like, I've always had a curious personality. I always wonder, why does this work in this way? Why does this even work? How does this work? If I do this, will this like make it stop working? Like, if I do this, will it help it to work more? Like, questions. And that's why I like science. Science brings conclusions, but it also opens up the door for more questions. Which is kind of the beauty about science. But that's my favorite thing about science. The fact that you get to answer things. You get to answer questions that are so complex. They're so complex that you can't really answer them unless you use some kind of rigorous approach. That's my favorite thing about science. Another thing is that you're accountable. So if you do this study in this particular lab and you find this particular thing, theoretically, if I follow your protocol, I should also find the same thing. If not, then it was not like a proper scientific approach. And you are accountable to not only the community of scientists, you are also accountable to the population that's going to absorb the science that you're like putting out, you know? So you're accountable to everyone. This is why you have to be like very strict, and not strict, but you have to be very focused. You have to be very you should record everything to like make sure you have a note of things. Like 5 years from now, you look back and like you know why you did what you did. Like you have a record of it.
That's the last one.
Okay, the last question is xenograft. What does that word mean? I never looked at what the definition of a xenograft is.
Xeno?
But I know what it is, but
Yeah.
I don't know like what's the Oxford definition of it.
What is
but
in your lab?
essentially in my lab, so my lab, as I said, we work on triple negative breast cancer, and in the past we've had mouse models where we have xenografted.
Yes.
In other words, we have placed breast cancer cells in the mammary fat pad of mice. And in that way, that's a xenograft. So there are breast cancer cells that we have in the lab, we place them into the mice. And then you can get the tissues, you can let the cells grow and what's not after X amount of weeks or months. You can then get that tissue harvested, and yeah, that is a process of xenografting.
Cool.
Yeah.
So that's how you give the mice the tissue that you're testing on later.
Are the cells that you're testing on. So specifically in our lab, how it relates is that we have normal cells with normal levels of KAISO, sorry, they're breast cancer cells with normal levels of KAISO, and then members of the lab were able to deplete KAISO, so they were able to take out KAISO out of the cells, and so we had basically two sets of mice that were injected with normal KAISO, mice that were injected with cells without KAISO, and that's how we were able to do our experiments. And basically what the lab found was that mice that were xenografted with the cells without KAISO, that the cancer was less likely to spread to other organs. So basically after injected it into the mammary fat pad of mice, which is like the breast of mice, the cells that were injected without KAISO did not spread to other organs, whereas the cells that were injected with normal levels of KAISO into the mammary fat pad, the tumors were found in, like the breast tumor spread to other parts of the mice, like the liver and the lungs specifically. So we're saying that KAISO is definitely playing a role in the ability of the cancer to spread. But I'm actually going to look up the definition of xenograft.
You're reaching for your electronics.
Xenograft, a tissue graft or organ transplant from a donor of a different species from the recipient.
Cool. Yeah,
Yeah.
xeno is like other.
Yes.
And then
graft is like graft.
Duh. Changing tissue. Yeah, I remember recently I listened to a TED talk about xenotransplantation. How they can get organ transplants from pigs.
Yeah.
Human organs that were grown in pigs. So that's like xeno, like a different, across a different species.
Yeah, so we have human breast cancer cells that were derived obviously from humans, and then they're xenografted into mice.
Yeah. How similar must we be if like the tissues can...
So essentially,
Yeah.
those mice were immunocompromised, and the fact that if they had strong immune systems, they probably wouldn't take up the cells as quickly as if they weren't immunocompromised. So that's something to also take into consideration. In most cases, if you're xenografting, it's most likely going to be an immunocompromised mouse. Yeah, but you must also be quite similar for the cells to actually still grow.
Yeah, because they're both mammals and creatures.
We're both
We're
creatures.
small and big.
We're both creatures. But yes, we are still genetically similar enough for the cells to grow. But I love that. We're still creatures.
Creatures, we're all creatures.
We're all creatures.
Thank you for having me.
Well, thank you for having me.
So thank you for sharing all about your research. It's cool because I have this much knowledge and Shawn has this much knowledge, and together we can learn a lot more. Yeah. So thank you for watching. As usual, be sure to hit that subscribe button for new videos every week. And remember that science is everywhere.
Okay, 1, 2, 3.
Science is everywhere.
How do eyes move independently?
Are you a chameleon?
I don't know, like you're the psychologist, like you must know no, you must not know. Not like you must know, but like you might know like how you can control different parts of your body that move together. You like look left, right, left, up, down, whatever, but to move one like independently. Like, some people can't like.
Interesting.
Yeah.
Some people can't do that, or even winking. Some people can't wink.
Yeah, I know people who can't. Is that genetic or is that
video idea?
More questions to be answered. This is why I love science. Like you can use a scientific approach to answer any weird question.
Science is everywhere.
Yeah, like you can answer any weird question. Be like, hey, how do people like move things?
Yeah.
Like
or,
individually.
Yeah.
How do people wink? Like
the other day someone asked why we yawn. And it turns out it's probably not a physiological thing like getting more oxygen. It's probably a social thing. Like indicating to others that you are tired, and then the other person indicates that they're tired. And it's like related to empathy and communication. Not like opening your mouth to breathe more.
I didn't think that. I just thought our brains were just like, okay, I'm tired.
Yeah. But then
I didn't think it was anything physiological.
Yeah, but if we're in the environment of evolutionary adaptation and we are tribal early humans roaming about, and then we have to decide when to stop and rest for the night, then it was like a form of communication. Maybe. Scientists don't know exactly.
Yeah.
But a 2010 meta review of yawning studies.
Wow, that's a thing. Wow.
Those yawning scientists.
Wow.
Are the ones they should interview next.
Yeah, I think you should do that.
Yeah.
I support this.
Yeah.
Okay.